So my name is Elizabeth Ross, and it's my honor to be this year's chair of the Scientific Program Committee that has assembled this annual meeting of the American Neurological Association. We've had a wonderful time here in St. Louis, and I have to say that the breadth and scope and interest of the presentations has been truly remarkable. We're all quite excited about it. Just to talk about a few of the highlights, we started with a wonderful pre-meeting symposium on the brain-computer interface, and the goals of that field is to find computerized ways of compensating for neurological deficits. And this is a new and burgeoning field that works both with creating brain implantables that then communicate to computers outside of the body or give a sort of closed loop interface in which there is communication that is worn by the patient and communicates to a computer. And this is being used for things like enabling motor function in patients with spinal cord injury or after stroke, and even can restore aspects of vision for patients with retinopathies or retinal diseases. And the approaches are all quite different, tailored to the neurological condition at hand. So in the area of motor function, we saw investigations where a sort of exoskeleton and an apparatus that connects to a patient's legs and trunk to enable an individual who is suffering from spinal cord injury or even brainstem injury to be able to think about moving, and that will communicate to the apparatus to actually make them walk. And this sounds rather Star Wars, but actually is coming to fruition and is really quite amazing. We saw an example of an implantable that is enabling a woman who's quadriplegic, who has no use of her hands or legs, to be able to think about lifting a cup and bring it to herself and to be able to drink with a straw. Truly amazing. And these capabilities are being miniaturized, and we're working with very fast microprocessing to make things more fluid and easier for the patient to use. We saw examples of communication devices that enable someone who's unable to speak, for example, an ALS patient, to be able to more rapidly type a communication so that you could actually have a conversation in real time, which is something that is enormously important for quality of life. And in one amazing presentation, we saw an example of using a specific capability called optogenetics, in which a virus that is carrying a specific receptor that's light sensitive can be injected into the retina. And by providing a light with essentially a type of glass on the patient, it's possible to encode that light signal in such a way that the brain receives the signal and can read it. So there's no implantable electrode, but it's a way that you can actually modify the stimulus coming in so that the brain can interpret what is presented. Truly amazing, very groundbreaking. And that was the start of a tremendous meeting. We had multiple presentations in our plenary sessions that went through problems such as Alzheimer's disease, both the autosomal dominant early onset type and the late onset type that affects a much larger population in this country. One of the things that we've been learning in the field is that while the autosomal dominant version that's inherited and usually caused by a single gene is certainly genetic, the late onset type also has a very prominent genetic influence. And through studies, for example, by investigators like Alison Goat and David Holtzman, it's becoming possible to understand what those risk factors actually are, and to have better tools for diagnosing and Alzheimer's cognitive impairment very early, so that we have an opportunity to intervene in an effective way. We also had a wonderful session on regenerative medicine, and this focused largely on conditions that impair learning and memory, and what we've learned about how to optimize the ability to recover after injury, infection, and any sort of neurological impairment. There have been great insights, for example, in the role not only of the microglia and the inflammatory process in cognitive disorders, but some insights as well into what the receptors may be that could be manipulated to improve learning and memory. One of the really exciting insights is that a particular cytokine that is working in response to the HIV virus and a receptor, CCR5, can actually be manipulated to improve learning and memory. An amazing insight that is actually going into clinical trials now. And I think the excitement of that meeting was, that plenary session was the opportunities that we now have to think about the period after a particular injury or during the process of degeneration, to be able to optimize the reparative processes in brain and spinal cord. We had a session on language processing that was really quite amazing. The difficulties that patients who are developing dementia can be very difficult in terms of their ability to communicate with those around them. And through understanding the different ways that language is processed and thinking about how one can optimize communication either by specific training or now with capabilities like transcranial stimulation, TMS, the opportunities for improving communication are becoming really quite amazing. We had another session on the microbiome that looks at the gut-brain axis. It, I think, was a proof of principle that was really quite amazing. Your grandmother probably told you that you are what you eat, and there's a great deal of truth in that, as we're finding neurological disease has an impact on the microbiome in the gut, and the microbiome itself has a tremendous impact on the progression of diseases like Parkinson's, multiple sclerosis, even stroke when it occurs, and that intervention with manipulations of the microbiome are proving potentially greatly effective in ameliorating the neurological symptoms of these disorders and slowing progression of disease. We had a session as well on clinical trial design, which is, I think, a real recognition that we've learned so much more over the last few years about what goes into a successful therapeutic trial. Many times we think that we know exactly what target to be hitting, and a drug is is created, and then when tested in humans, the results may be disappointing. And it's difficult for us to know exactly why that is. Was it the wrong target? Was it the wrong population who should be receiving the treatment? Were there improvements that we actually missed? So this session is talking about many of the features of successful clinical trial design that will accelerate advances in bringing scientific insights at the bench to the clinic and to patients. A really very important session. So it's just been a wonderful meeting. We have been working with people at all stages of their career trajectory, from students to residents and fellows to young faculty and senior mentors. They've all been interacting and sharing their experience, even among our chairs, and we've had guests from NIH talking with us as well, and I think everyone's leaving the meeting excited to go forward and investigate.